Yeah. So, so yes, very much. Yeah. So we begin now. It's a pleasure to have Dr. Amulya Panda. Um, he has a very interesting background in the sense that he is basically a biochemical engineering person from, did his BTech from HBTI Kanpur, the very well-known place, followed by his master from IIT Madras and Ph.D. from IIT Delhi under Professor Bisaria. And then he joined NII and he has spent last 31 years till last year he was there. And it is nice to have Amulya because he has seen the challenges of modern biotechnology as a young person, especially the inclusion bodies, which were the challenges of those days, if I remember right. And biochemical engineering was a integral part of any growth of biotechnology. And I'm glad that Amulya comes from a rich experience of having worked and seen people who were the founders of biochemical engineering in India, especially Professor T.K. Ghosh. And, today very, very fortunate that we'll be hearing him. Currently, Amulya, after having retired from NII as director, he is now associate director at Panacea. But it is always a pleasure to have you, Amulya. So now I hand over the mic to you. Please deliver what you want to talk to us about. Yeah.
Okay. Thank you very much. And indeed, I could not have asked better than meeting you all people. I still remember when I joined and you guys were, all of you, including, I go to Dr. Amrita Biswas and far beyond, Dr. Ghosh and of course Dr. Gandhi, Dr. John and other people were there, Dr. Bindude. But before I start talking, let me have a small disclaimer. Whatever I'll be talking as a personal capacity, and this is based on my own personal experience, which I had. I must say, wonderful experience for last 32 years I spent at NII campus, both in terms of my experience, both in terms of doing science, doing research, as well as living there happily. So these are very personal things, and this is my opinion. I hope many of you agree. If you don't agree, that's also fine, because I'll tell you what I felt during last 32 years. But let me tell you, I told also during once Professor Talwar came to inaugurate this the auditorium in his name. I was very fortunate because I was studying across the road in IIT Delhi when the NII was building up, Department of Biotechnology has built up. And I got a chance, I joined, and I never felt like going anywhere. That is the beauty of NII. I must say, those days in 1990, it was a privilege. I don't know. I got selected, I worked, I never looked back. But let me tell you the genesis of making this National Institute of Immunology. I wish I could have showed you some presentation, but when the land was allotted around 81, actually 81, Dr. Ghosh or maybe Dr. John or maybe Dr. Gandhi may or maybe Dr. Bindu they may be knowing, NII was set up in 1981 as an autonomous institute by Department of Science and Technology. That time there was no DBT. So in 1982-83, the real NII director started from All India Institute of Medical Science. Then they went to GK2, they lived there for 4 years. 1986, they came to the campus. In fact, 1986, when Department of Biotechnology was formed, it was taken up by DBT because it was the first autonomous institute, all you know. But having said that, I joined around 1990 when Professor Talwar was the director, and what a wonderful way of a man coming to an institute where, the only thing is you do something and deliver. It was the dream job to come. You come, work, stay, enjoyed. That was the beauty. And I think in last 35 years in that campus, NII has contributed, if you ask me, contributed primarily on three things. But before that, let me tell you why NII was set up. I think around 79, 80, the great people like Professor M.G.K. Menon, they thought immunology is a burning area and we should develop a immunology institute. That was decided by DST. I remember, I think Dr. Manju Sharma with M.G.K. Menon got it sanctioned from Mrs. Indira Gandhi those time. There was no cabinet committee, things like that, approved, and National Institute of Immunology was set up officially in 1982 in AIMS and things like that. And in those early days, I think we enjoyed a lot. Not that we didn't enjoy the latter part. The genesis of that building and the concept of National Institute of Immunology goes primarily to the director who thought those times how a modern institute to be built. You must appreciate Professor Talwar's concept. Today we are all talking of tech transfer, startups, product development. You know, in 1989, the then Drug Controller General inaugurated a product development lab in NII. Forget about basic research. That was the imagination of that person. I think his imagination of getting the best of talent, doing best and doing relevant science. In those times, he has only two objectives. Do research which is relevant to the country. And two things were relevant to his mind. One, controlling population, because that was his strategy. He wanted to develop a vaccine for population control, STG, and infectious disease. And he was too much to do relevant science because his whole objective was, let's solve the problem of India. Leprosy, tuberculosis, malaria, diagnosis, population, because nobody others will do. And do in an indigenous way. I think that is the genesis by which NII was set up and it carried out. And if I look back after living and working for that institute for 32 years, this is a dream come true. I mean, when I go and talk to people when I was working there or even now, I mean, if you to know what is NII, you must live there and you must understand. Everything you ask, it was there. I'll come to back why NII is NII, but let me tell you what NII has really contributed in 32 years. People ask what you have done. But I think the biggest thing what National Institute of Immunology has done is creating wonderful manpower. We have a PhD program. I was talking, we started the PhD program in immunology in 1986. Until today, we have 525 PhD students who have graduated from it. 500 PhD students. And if I take the students, what they have done exceedingly well, both in academia, both in industry, and everywhere. If you talk about academia, look at our students are all now on the clinical and immunology head of Johnson & Johnson, GSK, Novartis, Takeda, everywhere. And not only that, they are all full-fledged professor in Canada, in USA. And my last student, he was joined here, the person who is Jaram Mina, he just joined IIT BHU. In fact, there are a couple of students who are other faculty at IIT, other IIT. Apart from that, our students are faculty in many ICMR Institute, DBT Institute, IIT, ISR. I think one of the greatest contribution of NII is creating manpower for future. The other thing is the research legacy which we develop at NII. I'm sure many of you do when you are with Professor Talwar or anybody else. We have wonderfully done. You take any scientist from NII, whether it is today or 10 years back or 30 years back, you'll see a conglomeration of best talent of the country. I mean, there was a time you could count, it's a National Institute of Immunology, but you could count there are scientists who are engineers, who are doctors, who are physicists, who are chemists, who are biologists, who are pathologists. veterinary biologists, everybody was there. So you could talk. I think that diversity among the scientists, and each one of them, the crystallographer, whether they were in old or now, are the best in the country. The chemistry were the best in the country. The biologists, the cancer biologists, or the immunologists were best in the country. I think that is one aspect which NI has delivered, creating an ambience and creating an infrastructure to do world-class research while in India. The other thing of late, we have created really leaders, and we have created institutes. Like many of our faculty, many of my colleagues has become director of CSR Institute, director of DBT Institute, has gone to different places and lead industry. I think in terms of generating quality manpower, both for doing research, application, and deliverable, as well as leading industry. Even if you take Indian industry, we have our scientists, all are in best places, whether it is Reddy, Dabur, Kadila, many places. All our students are heading now. And I think that is one of the biggest contribution. And I must tell why NI was been able to contribute so nicely within the ambient of DBT. You must understand, I think the credit goes to many things which Professor Talwar started. And if you ask me why NI is so good, why NI has achieved so big things in terms of many things, and why everybody envy, if I am allowed to say envy NI, I think because of two main reasons. One, the science we do, and number two, the infrastructure and ambience we provided. And both credit goes to all the people. I'll talk about the science policy, then I come to things. See, the best thing that has happened in NI in last 50 years, which Professor Talwar started, is there is no hierarchy among scientists. I think that is the best thing NI could offer. Anybody who join is independent. He directly report to the director, to SAC, and he is independent. There is no official burden. I have to go through this place, like in many places. The independency to the scientist, I think this is the biggest requirement. NI is NI because we all, whoever joined, is independent. Look, for me, if I take my example, when I joined from IIT. People thought, what an engineer will do. But because I did well, I could take the position of director. That is the ambience NI provides. I think this is the model research institute where there is only one thing: your quality and your productivity defines your status. No hush-push, no administrative difference. I think the best thing NI which has provided is providing independent to the scientists, and the way we pick up scientists and students. Till today, we have a very rigorous way of picking scientists. Remember, we are in Delhi, we don't get into any other business when we pick up our faculty and students. I think that is the second reason. We pick up the best faculty in the world through a rigorous process, no back pushing or anything. In the same way, we pick up our students. Every year, we take interview of nearly 3,000 students and pick up 20, the best of brain. I think most of the things in NI is because of the faculty, the independence of faculty, and the students and the faculty which we recruit. But more than that, what has helped NII in scientific terms in the last 30 years is the rigor in which we are evaluated. Our RAPSAC, our international review are too much. I remember when we were young, during RAPSAC time, we were really used to be scared. We used to sweat. You go and talk, before you start talking, there are doyens sitting there. Everybody, all, Professor Ganguly or Bijayan or Padmanabhan or Surolia, so many people. Once you, or Professor Tandon, once you face them, you are tested. I think that RAPSAC, the rigorous monitoring and evaluation, and international screening. So scientifically, giving basic freedom to the scientist, doing recruiting wonderful human being as a faculty and student, and evaluating through a proper RAPSAC mechanism was the real beauty of this NI science which took place. Apart from that, there are other things which also help. The other thing is we had an unwritten rule. Nothing belongs to anybody. All equipment are in suits. Even we have an unwritten rule. If any faculty denies other faculty not to use equipment, the director has the power to take out that equipment from that lab and keep it in the corridor. There is no personal belonging. We all used to share. That is one good thing. The other thing is we have wonderful and less minimal administrative support. Our administration was small, whether from a senior manager to the finance to our support staff, all were wonderful cooperative. I don't know how that culture developed, maybe with Professor Talwar or subsequent things. So apart from science, the infrastructure which was built up, we had obviously the funding from DBT very lavishly to set up wonderful infrastructure in terms of equipment, in terms of animal house, and the administrative staff. But most importantly, I think the sustainability of NI's supremacy was because of its wonderful directors over the period of time. Think carefully. When Professor Talwar was there, he only stood for doing relevant science. He has to do something deliverable for the country, whether it is a population vaccine or something for leprosy. After Talwar came, when the things was settled, came Dr. Basu. He has only one objective: do fundamental science, do excellent science. So from relevant science, the NI became accustomed to do relevant, really fundamental good science. Professor Basu was only on impact factor, which was many may or not like, but when you want to be competitive, that is needed. After Dr. Basu came Dr. Surolia. He was only interested in doing innovative science. He has an applied mind, do patenting, do things. So starting from Professor Talwar to Surolia, from a relevant time to excellent science to innovative science. Then came Dr. Chandrima Saha. She started a different culture. She started science with a human touch. Very wonderfully talking to the people and disturbing them. And when I came, my term came as a director, my job was very simple. I don't have to do anything new. I just pick up some quality of Professor Talwar, some quality of Dr. Basu, some quality of Surolia, and some quality of Dr. Saha. My job was done. So it was a blessing that over the period of time, NI has seen wonderful directors who infused different ideas which was need of the time. See, when it was starting, the need was to stabilize. Professor Talwar wanted to do and show the world in a minimum time we can make diagnostic, we can make vaccine. He did leprosy vaccine. Then come the period of sustainability when Dr. Basu came. He showed, okay, publish paper, be good in your science, we do excellent science. Then came Surolia, then came me, and I am happy the next person was come. He is also from a physics background, and I hope that will continue. So one of the beauty that has happened to NI is getting a relevant and excellent director at the helm of affairs to take NI to a different facet. I think that was important. So because of these three reasons, because of no hierarchy, having wonderful infrastructure, beautiful administrative staff, and lavish funding from DBT, along with very experienced director, that's where NI stand. And I think that's why NI is a great model to replicate, to do biological research in the country. I think I'll stop here. If you want to ask anything about how NI or what others do, I'll be very happy to answer questions. Professor Kanan.
So thank you. Yeah, please, Bindu, please go ahead.
No,
Yeah.
please. No, I had a few questions.
Yeah,
You go
please.
ahead, then I will ask.
No, you ask, Bindu. I can ask later. No problem.
Amulya, you have to tell some interesting things about NII because I was the nodal officer of NII for about 14 years.
Oh, yes, I forgot.
You have to tell the story of Dr. Talwar getting, trees planted at a particular dollar rate by each of the visiting person coming from outside. That
Oh
was a
yes.
very interesting aspect. Then I would like to mention here that one of the scientists is of NII is now the secretary DBT. Uh,
Yeah.
that's a very important impact that you are coming from an autonomous institutions and now you are heading a very beautiful organization named Department of Biotechnology. And Dr. Amulya, would it be time consuming if you briefly touch upon the Mycobacterium W story,
Yeah.
or little bit about how cancer sciences suddenly came up in your this thing, and your own protein purification efforts, and its impact on the industrial framework, because these were all very successful stories of NII in translational terms.
Yeah, the Mycobacterium story I can tell because when I joined, first thing I was told, grow this Mycobacterium in fermenter. You remember, many of you know, Mycobacterium is microaerophilic. If you give air, it doesn't grow. And its lipid structure basically determine its antigenicity. And so we took time and we could grow. We could see, me and my colleague, Ashok Mukhopadhyay, both we could grow, and that was the time we grow it in fermenter. We made huge number of doses, but Professor Talwar wanted to know the one which we are growing, is it really protecting? So you remember that time I am an engineer, Ashok Mukhopadhyay was also engineer. We did an animal immunized study and we took out the spleen. We didn't know how it looks, but finally they confirmed, oh, this is protecting. And that time I remember for a Chengalpet trial, we have a product development cell which was GMP certified in those days. We made more than 5,000 doses for Chengalpet vaccine trial. So this was the story of Mycobacterium to begin with, which was working on leprosy. That time the trial was going on Kanpur there. People were clear. But over the years, Professor then again refounded, still is working, that this Mycobacterium W is atypical Mycobacterium, like incomplete French adjuvant. So this can be used as an adjuvant, as an immunomodulator. So he tried using many vaccines, like with rabies and cancer. So basically, this organism has a potential to activate the immune system, and particularly for a disease where you have a immunosuppressive environment, this helps. In fact, this was lastly Sangeeta and others, they could show that this works on MDR TB, and it was promoted as a TB vaccine. So this is a very fascinating thing because it has all discovered, developed and formulated, and now it is with Cadila they sell. So this is an interesting story of Mycobacterium W. When this was going on, slowly people came and they started working on tuberculosis, malaria, and cancer biology. So this was used. Sangeeta has used this as a adjuvant for adjuvant therapy along with Taxol and other things. So this is how things go. If you ask me how this was on those old days, there is a lot of excitement. We are all young, and we have no restriction of getting any fund because Professor Talwar was, because we are the only, that time only NCCS was coming up, so we are the only alone autonomous child of DBT, very pampered, getting all sort of things, all project. And we have a wonderful time because with Professor Talwar, I was just discussing before evening, we have every week wonderful people coming. One Nobel laureate coming someday. You see, I see it for the first time. I saw in 1990s Nobel laureate coming, JRD Tata coming, or even many people come, many eminent people coming, giving talk and things. And the living was wonderful. The campus is too good to leave. This is the best thing I could get to live in the campus for 32 years. You have everything. You have, I don't know, many of you whether know it or not, in the initial days, all the residents were air conditioned. I remember my first 2 year, I was just living in an air conditioned apartment. Go to the lab, come and leave. And telephone, every facility was there. So it is the ambience and the infrastructure and a vision which Professor Talwar started, which continued with successive directors, that helped in the National Institute of Immunology to build up as a primary institute. I remember particularly for my work when I started, because I came to grow these vaccines in the fermenter, then I started growing recombinant protein. And when you grow recombinant protein, you end up having inclusion body. And it was challenging. So almost around 98, 99, I started working. Touchwood, I work, and I'm happy that the protocol we developed without using urea guanidine is now very extensively followed all over the world for recovering inclusion body therapeutic protein. I'll stop here.
Amulya,
Yes, sir.
when you look at the early background of Professor Talwar, how did he become an immunologist?
I think, you see, I tell you, this is a personal thing. I mean, how big he was basically a chemical technologist from
Correct.
Lahore, and he did his PhD was within some receptors and hormones, and then he went to Institute Pasteur, got an exposure, and when he came to AIMS, he set up a WHO Access Center for Immunological Reagents. I think the interest started coming there, and then he picked up from the hormone. He picked up the beta HCG, and then that novel idea, if beta HCG is the protein, develop an antibody against beta HCG, that will be a vaccine. So that's how we enter. I think I tell you, these are the people who did actually translation in their real time. In those times, somebody doing chemistry and chemical technology coming to do immunology is a real challenge. I'll give you another example. As I told you, Professor T.K. Ghosh, I remember him. He did chemical engineering in 1942 from Jadavpur. And can you imagine, in 1942, he went to do Ph.D. in microbiology? These are the people who are real translational people. Same thing with Dr. Sujoy Guha. He was an electrical engineer, did PhD on electrophysiology, studied medicine, and became a translational. I think these things, these 3-4 people, I can name a couple of people, like many of them, but these are the people who changed their boundary in the early days. Remember in 1940-50, where things were not stabilized, we are still coming up as a country, just independent, no infrastructure. But in those times, their vision to do something different leads to NII or biochemical engineering by Professor Ghosh, or maybe the biomedical engineering by Sujoy Guha. I am fortunate, I know all 3 of them personally because I studied in IIT, got a chance to work with Professor Talwar. So this is the kind of things people do, really translation. I'll tell, these are the real heroes of Indian translational biology.
I think you made a very valid point that even though they were from a different background, but they knew what was relevant for the nation.
Yes, for those times.
And they were able to shift. In fact, I would request Professor Muralidhar. Are you there, Muralidhar?
Yeah, I'm there.
Yeah, can you just elaborate, because you are a hardcore immunologist of those days, and can you share some of your thoughts on how immunology as a subject developed in India?
Well, I don't know if I open my mouth on this, but just for this audience, I will tell only the non-political things. See, immunology as a subject did not develop in this country. It was essentially witchcraft. Only medical colleges had a paper in immunology. However, some people like the JNU group, Madurai group, they started really cellular immunology in the real sense. Some of us entered immunology through backdoor because of chemistry background, and so we were practicing only immunochemistry. Slowly it became part of the curriculum, I was the first one to introduce immunology in MSc courses in this country, Delhi University. Leave that, we'll come back to this pregnancy vaccine, which Talwar championed. And somebody was asking how come he got interested in this. A small correction, he had a diploma in leather technology.
Yes, chemical technology, leather technology were there.
Yeah. And he got his position in All India Institute because of his aunt, Rajkumari Amrit Kaur, first health minister. Because he was in biochemistry department, he was moving around in the Guha Research Conference. And in 1973, there was a big international symposium in Moudgal's lab in Bangalore, Indian Institute of Science, where breakthrough research was announced that if you use luteinizing hormone as an immunogen, you can induce sterility. That means pregnancy can be blocked, ovulation can be blocked. All such things were shown, and it became a world famous work. In that international meeting, two stalwarts came, one G.P. Talwar from Delhi, and another, and here I will reveal something which most of you might not know, that in Punjab University, Lahore, originally it was in Lahore, it is still there. Later it shifted to Hoshiarpur when we got independence, and then shifted to Chandigarh. 4 people were MSc classmates: Hargovind Khurana, G.P. Talwar, Om Prakash Bahal, and Gurbakh Singh. 1 hour back, I was attending a Khurana Memorial lecture by Raj Bhandari, and he showed me the early papers of Khurana from Lahore as a postgraduate student, along with Gurbakh Singh and others. Now, among these 4, the story of Khurana is very clear. As you know, he went to Manchester, he went to Prague, he went to Canada, he went to Wisconsin, he went to MIT, Nobel Prize, all that is a very glamorous thing. Om Prakash Bahal, another classmate, after serving as a school teacher, went abroad, got his Ph.D. in carbohydrate chemistry. Then he went to California as a postdoc and picked up protein chemistry from Smith. And then he was looking for a job, and he got it in University of Buffalo, SUNY Buffalo. And the head of the department was one who worked on amino acids, amino sugars. And at that point, this man advised him, instead of both carbohydrate chemistry and protein chemistry, don't work on a glycoprotein which is unimportant. Take a very important one like a hormone. That's how he picked up HCG. And his tremendous work on HCG drew the attention of his classmate, Talwar. And Talwar got influenced at the symposium in Bangalore, where people were talking on immunocontraception, much before. It's all Bangalore work. And then, but he was a very clever fellow, very intelligent. So instead of working on pituitary hormone, which affects menstrual cycle, very cleverly he picked up HCG. And he didn't have experience in protein purification and other. So he collaborated with Om Prakash Bahal. Bahal supplied him HCG subunits and all. The rest of it, every one of you know. There's a lot to talk about. More than 500 papers came on immunocontraception from these 3 groups, Adiga, Moudgal, Talwar. But Talwar has a great leadership. He was very lucky. Swaminathan advised on the establishment of NII. He got it. Very imaginative leadership. Look at the building. The other things which Panda has listed, this creative ambience, atmosphere, infrastructure, living on the campus, facilities, everything with very little administrative interference. To that, you should also congratulate Bose. He was the first manager administration. In fact, when I asked him, Talwar, why don't call him administrative officer? He said, then head will swell. So I call him manager administration, and he's an MBA. That's
He is senior manager.
yeah. Whatever it is, a lot of good things happened. I was also associated with NII in the RAC meetings for a long time. But it's a great achievement, and we should thank DBT for generous funding when
Oh, yes.
other institutes were not there to compete. Later, of course, NCCS and many others came. But in that way, a bit of luck, but tremendous leadership, tremendous technical talent, all that made NII one of the best institutes in this country.
That's very interesting. You know.
He was also Talwar when he was in Paris. His roommate was Jamshid Tata, whom you mentioned. I worked with Tata, I worked with Talwar, I worked with Ball, I worked with all these guys.
All big guys used to come to NI. And,
Yeah. It's a great story.
Now, Panda, I would like to ask you that you talked so much about Talwar and his contribution. Now, what has been the significant contribution which may have changed the direction of NII with every new director taking over? Did any major changes happen on the focus?
No, see, maybe over the years, because when the contraceptive vaccine was not showing good, we changed our focus to modern biology, doing less, but it was according to the tune and requirement of the science that was developing. I wish we could have done exclusively only on immunology, because if you see immunology now, there are 20 different varieties of T cell and innate immunity has come. We lack little bit, but as I told you, there may be a different change, but please remember the NI is a very small institute. The capacity is small. It is very intense. So after Talwar, when Dr. Basu came, his objective was make your foundation strong. I mean, good impact factor. I remember I was an engineer. I was not publishing. He said, world only know scientists who publish. What are you doing? So I start publishing. Fortunately, because of my engineering background, I start publishing in biomaterial. Biomaterial impact factor is 9. I don't blame it, but I get away and I publish 6, 7 of them. And I really take credit of publishing. So I must give credit to the after Talwar, Professor Talwar, Dr. Basu. He groomed scientists, put a truthfulness and fundamental, that inquisitiveness. I think that was essential. The initial phase, as Professor Muralidhar said, was to stabilize the institute. A new institute is coming, new concept is coming in India, those 80s, 90s. You have to develop. Once that is stabilized, you have to make it strong. I think Dr. Basu did that. He infused, do research, do fundamental things. I remember Dr. Bindu Dey was here. He said, after studying from IIT, what are you doing? Go to the best engineering college. And I applied for a Indo-US fellowship, which Dr. Bindu Dey was handling. And I went to Berkeley. That was the things he did to me and he did to everybody. So that sort of grooming we are all young. He wanted that we should be strong enough. Of course, he was too much towards science, but I think that was the need of the day, because after 10 years, we feel so strong, nobody could touch us. Like once I start publishing in the best engineering journal, my confidence grew. It's not only me, all my colleagues. So after Professor Talwar, when Dr. Basu came, he strengthened us with fundamental science. Your core competency is improved, and that was needed. That was the time we started publishing good paper, good journal, JBC, Cell, Immunity, all these things came. It became a normal to us. Then came Professor Surolia. He was part of this. He wanted whatever science you do, it should be innovative. It should be a lot of patents, a lot of industry, a lot of application. And then came Dr. Chandrima Saha, taking care of all these things, because you see, by the time Dr. Chandrima Saha came or I came, we are 40 years old. Things have become old. People who are old, they have different perspective. You have to take care of your family, you have to do wild science. So she has a very human touch on overall things, doing everything, same thing. So over a period of time, no doubt, the immunology was a little bit eroded, but probably it was replaced by doing fundamental science on cancer biology, on aging, on TB, malaria, very, very fundamental work. That strengthened, we could do very basic world-class work, publishing in EMBO, in Nature Communication. That gives us a strength. I think even though the broad objective of immunology is slightly deviated, it was in the proper perspective, I'll say. And when I came, I started, because I am an engineer, I started an immunoengineering division where you could grow T cell, B cell, CAR T cell, immunotherapy. I hope that continue. And now it is the area of immunophysics, artificial immune system, artificial antigen presenting cell, system biology, system immunology. And I'm happy that a person with the physics background has become a director. I think I will tell at different stages of NII, good fundamental people with different outlook, different background has come, and that helps in all together without losing the path. See, we still don't compromise on the quality of research, basic things, and collaborate with each other. I think that way it has done well. Nothing much changed, but towards a goal of developing a strong foundation-based National Institute of Immunology.
I just want to ask just one question, that when you look at COVID, for example, what is the kind of role NII played in the management of COVID?
Okay. So during, in fact, during COVID time, I was the director because it was a certain things, and we are not getting a lot of samples in hospital because it was from hospital, and in NII over the year we have very little human immunology or things. There are 2-3 people who were working, they were involved. So what we did, we started coding, like my group was making, because we were making inclusion body, we made a RBD protein in E. coli. Now nobody has made that in E. coli. We express, check its immunogenicity. But more than that, what we realized, we didn't do a lot of assay, but we realized that across the country, if you go to small place in Odisha, Bihar, West Bengal, or UP, these MLTs who do assay of blood glucose, they're not exposed to do RT-PCR assay, like COVID thing we're doing. So we started with PSA office. Dr. Vijayaraghavan was very helpful. He gave us fund. We started training of this MLT of medical college from, I remember we started a little late, but every month we have a 30 days class. 1 week experiment, 1 week on PCR, 1 week on pipetting, 1 week on BSL-3. So that was one of the contributions where we have trained maybe 300, 400 government employees across Bihar, Odisha, West Bengal, UP, so that they not only know RT-PCR, they know other modern techniques. They were all exposed, but that is one of the things. Plus, we did a lot of assays, and we did develop knockout mouse, many things.
Dr. Panda, can you people hear me?
Yeah,
Yes.
we can hear you, sir. Yeah.
Dr. Muralidhar definitely has given us the background on beta HCG, and Dr. Kannan also has raised a question regarding priority and all that. See, these programs, we were handling these programs, and I was with me, and I was also taking care of the international collaboration. This particular vaccine, HCG vaccine, reached to a stage where it could have gone further. Not political thing. Inside, what happened that it could not be taken further because so much money had gone. I think by that time, if I remember, more than 23, 24 crores had already gone into this process. But it could not be taken, So that concerned, me as responsible for, this NII or, looking at the international situation. Can you just give us the insight, what you people thought about that?
Okay. No, I know this is a very difficult question, but
Yeah,
the point. No, let me tell you. Let me give a
Nothing personal. It's
no. This is
It's a very difficult question to answer, Amulya, but I didn't want to ask. It's good that Dr.
no. I
Gandhi
can
has
answer.
raised that.
No issue. No malice spoken. It's a very valid question. I think during my talk somehow I forgot one of the contribution on the growth of NII was the international collaboration. I'm sorry, I forgot it. And Dr. Gandhi was doing there. During those early days, we could see people coming from NIH,
and all that.
everybody. And I could see Professor Talwar giving personal attention to the, like Barry Bloom or many of them, all Nobel laureate coming, and Professor Talwar will give personal interest. Has he come? Is he staying in the guest house? Has he taken food? Has he gone there? Apart from giving a lecture. We are so thrilled. But coming back to SCG, I think by the time the second phase of SCG titer was coming up in the clinical trial, that time Professor Talwar retired.
Yeah.
And then there was a shift. Those old people retired from, even DBT also was not, if I am wrong, supporting that this should continue, because we are at a phase 2. We couldn't do the phase 3 because that time this has to be produced in a recombinant way. At that time, I think, I don't know whom to blame, but please remember that time the new, let me tell you, that time the new regulation has come. And Professor Dr. Ghosh is there. For a new regulation, a company has to produce in a GMP. When Professor Talwar started, even leprosy, you have a good intention, you make in a GMP lab, in the lab. Our regulatory body was not that strong. We are evolving. We could go away. But that time, the new regulation come, you have to make in a GMP pathway. You have to do preclinical talks, phase 1, phase 2. I think that was one of the building block of blocking that unless some company takes it and make it and produce in a GMP way and do preclinical, it cannot go through. I think that plus the interest shifted because of other regions.
See, that would not have been difficult. That would not have been difficult at that time, because once we had reached to that stage, GMP manufacturing could not have been a problem. So many people could have got interested.
It is not SCG. It is, if I may be allowed to say so, it is not about NII or anything. I have seen in many places, we become too personal. SCG, Professor Talwar ka hai, ye inclusion body Panda ka. We must see that science belongs to the country. Country has put money. It should be independent of any person. Once he or she who retires, evaluate that science. If it is good, country has put money, should have continued. I'll give you another example. Apart from SCG, another area which Dr. Gupta did is Jona pellucida. He is number 2 in the world on Jona pellucida glycoprotein. It is not his protein. I tried my best. I wish things could have continued at least for the sake of science itself. But unfortunately, it's not in NI. In many places, when the director changes, the idea changes, the vision changes, the perspective changes, I think one of the requirements for a long-term objective of the country is the sustainability of the program. That should be encouraged. Somewhere, as a country as a whole, we miss that. This is my perception.
Okay.
Karna, can I butt in?
Yeah. Sure, Muralidhar, please.
I think this is an extremely exciting point that we are just discussing. There are 3 aspects of, because very embarrassing question was raised, what is the status of this vaccine? There are 3 aspects. I will leave out and I will not talk about the political aspects. We can speak for hours together. Leave all that. But there are 2 important things: the changing policies from the funding agencies, including WHO. When there was a little bit of cross-reaction with pituitary LH in Talwar's vaccine, we know that 90% of amino acid chemistry of HCG beta is identical to or similar to human LH. And there was therefore a cross-reaction, and therefore noise was made that cross-reaction means menstrual cycle will be affected and so this will not be acceptable. WHO played lot of games. I will not go into detail. I know everybody who made this into a mess. They simply dropped Talwar like a hot brick. Anybody can question me. There was a mention of this zona pellucida. I remember, Dr. Gandhi, I accompanied you to Washington in the Indo-US meeting. And you remember there was a project from NII. It was rejected because people are those who were immunized with the zona ZP2, ZP1 and all. There was a slight cross reaction, and therefore some of these animals which are, it was acceptable to Catholic countries because it was called as a true contraception, prevention of conception. But in those animal models where it was immunized, they showed oophorytis as a symptom. And the American side, in spite of my strong talk, rejected this project. This, I would say, is a political play by funding agencies. WHO, for example, there was a guy in Ohio called Vernon Stevens. He proposed a candidate vaccine based on the C-terminal 35 amino acids of HCG beta. And everybody in the field thought it's a brilliant idea because human LH does not have these 35 amino acids. So in one go, there will be no cross-reaction. Therefore, no menstrual cycle will get affected. Patients will be psychologically good. Therefore, WHO simply dropped Talwar and supported Stevens.
That was unfortunate.
$300 or $30 million were given to him. However, those of us who have done the work also on the sideline, we realized long back that antibodies to C-terminal peptide do not have bio-neutralization capacity. Stevens never mentioned this. WHO did not have the brain to analyze his data. Finally, this story came out. They were non-neutralizing antibodies, therefore the worst candidate for pregnancy vaccine. By the time, you hurt Indian groups, Talwar and other people, and therefore there was a delay in all these things. There are a lot of technical problems here. I will not go into detail and all that, but one thing people should appreciate, there was also a semi-political movement in this country by scientists to belittle the entire work on reproductive biology in this country and say that this entire funding of pregnancy vaccine is a waste, it is a bottomless sink, all sorts of things. Talwar was targeted to take him off from funding. Ramalinga Swami, being a very neutral person.
We all know, sir. Let's
Sir,
discuss that. And I think
Sir, we didn't want to discuss it because,
No, but it is important also to know the background of
Why
how
did not
games
it work?
people play. Yeah.
no, sir, it's not
Dr.
about
Musa.
SCG. It's many places in the country.
We are
The
going
science is
in
never
a
respected.
different
The
direction.
person is respected.
You see, I want to say something.
Yeah, sure.
You see,
Go, sir.
first of all, you have to realize that this webinar is about knowing the contributions of NII. It is not about knowing Professor Talwar and his criticism. That is not the intention of the
Absolutely right. That's why I said
Let
we
me
will not talk
complete.
politics
Let
here.
me complete. When I was a DBT officer, I have plenty of opportunities I had to interact with the NII scientists, starting from Professor Talwar up to Professor Surolia. And of course, I knew Amulya very well because I am a chemical engineer and he is also a chemical engineer. So our interactions matched quite well. The point that is to be remembered when we discuss NII, which was basically a DST institute, later on given to DBT as a biological institute to produce biological substances, we thought from the DBT that it will make substantial contribution in terms of applications in biology. What happened is what I found till I was in DBT up to May 2002, the NII had concentrated more on understanding the basic traits of how biology is working. Biology and biochemistry are very well connected area. There was very little emphasis on in the engineering of the biology, and therefore the engineers who had to produce recombinant DNA products like Amulya Panda, they did not come into sharp focus. Actually, what we wanted or we desired as administrators from the DBT was that the National Institute of Immunology will come on the forefront in the development of application-oriented vaccines. Actually, one vaccine came out quite successfully from there, which was Professor Talwar's AMW vaccine. And this vaccine strain, I had uh, great association with it because I had collected this particular strain from the NII when I retired and took it to Cadilla, where I was initially a consultant and later on I worked there as a working scientist, became a president later. I found that we had produced a kind of suspension with MW, and but that suspension was not stable. If you keep the suspension for a while, it would get into two layers. So from the Cadilla pharmaceutical, since we had a background of amphiphilic substances, I put some amphiphilic substance in it. Two particular things we put. One was 2 in 80, and the other one I do not want to name because this is a very important one we used to use in HAL. And we found that the product is becoming stable. That is how we improved upon the vaccine. This vaccine actually now is used as a immunotherapy in improving the presentation of the antigens by the dendritic cells to the T cells, and thereby it imparts in increasing the Th1 pathway of persons who take this vaccine. So this, I must say, is a very positive contribution emanating from the work of NII. This is one thing I must say. The other thing that we actually thought will come out was that much talk has been made about this HCG vaccine. At one time, I and late Mr. Biswas, advisor at that time of DBT, had written a review. We did not publish it, but it was written. We found that basically the difficulty that was happening was the presentation of the beta HCG to the immune cells was not very properly understood. We could not get a very good adjuvant which could do the job. Professor Talwar actually conjugated the beta HCG with cholera toxin. He also conjugated with I think tetanus toxin.
Tetanus toxin.
And he tried, but then the complexity of playing with producing antibodies to a hormone which is essential for life process was not probably well understood. But that doesn't matter because naturally we understand science little by little and we make step-by-step process. But then the contributions that were made in the development of HCG, anti-HCG or anti-HCG vaccine to contain the population control, which was actually supported by late Mrs. Indira Gandhi and later on was tried very practically at NII. It was certainly a good work, which although did not lead to any product, but then it certainly gave us great opportunity to understand how actually things can go wrong when you tinkle with hormones which are essential in the life process. So I would not go beyond this on NII. I must say that I have seen great scientific work that has emanated from NII. We have made tremendous international name by the publications in the basic scientific area in a lot of good journals. Of course, in any work, ultimately nothing success like success. And in that context, I must say that only one vaccine, that means MW vaccine, was the creation of NII, while we expected many. Our Dr. Krishnamurthy made a very valid question on what was the contribution of NII during the difficult COVID-19 days. We really, as outsider from NII, found that much more contribution was made by several other institutions of ICMR than many other institutes of DBT. Actually, the great work of DBT, which is many times not mentioned, is that they created facilities for genomic sequencing of the virus, and this gave us tremendous lead on understanding how things are changing. We thought that at that time, during that COVID-19 period from December 2019 to the present time, some new vaccine lead will come, but from NII. But that has unfortunately not come. Thank you.
I think.
Can
I
I
think
add one sentence?
Professor Mullah. Yes, please do.
I think, see, the focus is on the greatness of NII, tremendous contribution. It has changed the culture of modern science, CCMB and NII. For that, hats off. And the biggest contribution came because of the leadership of Talwar. Even the pregnancy vaccine, of which so much politics came, it would have succeeded. Amulya Panda mentioned about the GMP facility and the recombinant protein being the product profile. Even for that, Talwar recruited many people. DBT also gave 4 or 5 projects to outsiders, but none of them produced recombinant HCG. So I would say that Indians let Talwar down. Otherwise, it would have been one of the most successful stories in immunology or immunoreproduction. We should salute Talwar and the kind of people he built up. I would only blame immaturity of Indian science.
That's also
I think it is a very fruitful discussion, because the aim of this thing is not, let us look at where we could have gone wrong, why we didn't support. These are all basic issues. Our country is well known that we don't know how to pick up a good lead and give a full-hearted support to succeed. I think this is the lesson I always find, that we have missed boats where we had the strength. And I remember Professor Muralidhar saying, Indian immunology was its peak, as he rightly said. We had such stalwarts in the country, and we were, as a country, we are reckoned in the international scientific community. Am I right, Professor Muralidhar?
Yeah. You see, the Journal of Reproductive Immunology is only fed from India. Most of the papers are from here. Later, America picked up all those things.
So I think whatever said and done, NII has played a critical role. Mind you, it was created during DST time and it became part of DBT. But I think overall, what I am very impressed about NII is many of the faculties have been publishing in Science or Nature, which has been consistently happening. I think that speaks volume of the quality of science which is happening in NII. I think for that I must compliment the way good people have been recruited and who sustain that quality of work. I think that is a very important one. And number two, as Panda rightly said, they have produced more than 500 PhD students. It's not a small number. Mind you, for
quality students, all very good, high quality students.
So that means you created a good skill development program, if I may say. In the last 40-45 years, I think you have an excellent skill development program which has come up. And I think that is what the government is now talking about, skill development. I think NII was much ahead of time at that time. Only now we are able to appreciate that how important these are. If there are any other questions, I leave it open.
I will just add one thing. Please remember, during Professor Talwar time till today, we have the best animal house facility in the country.
Very true. Very
All
true.
animal, transgenic, and non-human primate center sitting in Delhi.
Yes.
These are unbelievable facility which NII has built up, maintaining, sustaining, and allowing others to use to do their experiment. That's the beauty of it.
I agree. I totally agree. Any other questions or any other comments anybody would like to add?
I would like to add just, a line or two. Amulya, you must add here that how NII has, emerged as a big brother to all other DBT
I
institutions.
told
Because in my time, I would always call that, whatever the manpower recruitment policy has come up, in NII, all other institutions became clone of NII. So they wanted to do whatever NII was doing. So much,
in
such
fact,
high impact.
Dr. Muralidhar told all credit goes to the administrative acumen of Mr. Bose, who used to call him Bose Saab. He knew everything. He knew rule, but while following the rule, he knew how to get the work done. That is the beauty of his administrative acumen.
Yes, yes. Yeah, partly, but there are many other reasons. But I
Yes,
would
yes.
definitely give marks. Yeah, sure. Okay.
Only thing I was a bit disappointed that NII, I don't know whether they contributed anything to the hematopoietic stem cell research.
Yeah, in fact, if you see one of my colleague, Ashok Mukhopadhyay, who worked with Bharat Agrawal, he has a wonderful setup, and he has done beautiful work. He has published in Journal of Hepatology. Can you imagine? Stem cell, and the Pramod Upadhyay, he works on retinoacid pigmentasa. He convert antigen presenting cells from stem cell, many things. But I believe now DBT has an institute of stem cell things. So more focus goes there. We more concentrate on immunology and other things.
Okay. If there are no more questions, I just would like to, what would you like to summarize your last 32 years in NII? What is the message you would like to give to young people?
Okay. I mean, for me, it was a wonderful time. I must say, I was blessed to get a job and work in NI for last 32 years. I have learned one lesson from NI in my lifetime. If you want to live not from a scientific, you have to make your innate and adaptive immunity both strong. When I say innate immunity, I'm not talking about dendritic cell or a signal. Innate immunity is your value, your culture, your integrity, your well-being. That is what we learn from our things. And adaptive immunity is your expertise, your qualification, your strength. So your integrity has to be high, your adaptive skills should be high if you want to live in the world. I think these are the two things I have learned. I have learned in a very tough way and nice way. But to put it in a simple way, make your innate immunity strong, make yourself, your integrity and your simplicity or your law of life should be strong, and your adaptability should be enough so that you handle. These are the two life learning lessons. And I must tell you, among these, all during COVID, your innate immunity is much better than your adaptive immunity. It's same thing. You have a competency and you have integrity. If you don't are competent, your integrity can take care of your competency. But if you don't have integrity, your competency cannot take your integrity. So at the end of the day, it is the innate immunity or the integrity of your scientist that is important. As long as you preserve that, the competency can be comparable, can be up and down depending on who is what. I'm an engineer, somebody is a scientist, somebody is a doctor, somebody is a chemist. The competency vary, but innate immunity, your integrity, your lifestyle, your culture, your well-being, that is what carry you further. That is the lesson I learned. And I'm sure during COVID, you all know, if your innate immunity is strong, forget about any vaccine, which work or doesn't work, we don't know. But if you are strong, if you have done properly through your childhood, taken care of all your grandmother's advice, you are okay with COVID. I think this is the innate immunity. This is what I have my lesson from my life at NII. Thank you very much.
So thank you so much, Dr. Amulya, for sparing a valuable time and sharing. And
Thank you.
I really admire all the participants who contributed also to enlighten us on various aspects. And I think it is a very healthy presentation because we knew much more and what are the challenges of doing science in India and how to propagate an idea. What are the challenges one faces? Even if you are dynamic, the system has its own way of balancing. So some things don't come out, some things do come out. But I think overall, I think NII creation has been, a milestone as far as government of India is concerned, because we have a strength and it has produced such outstanding scientists who continue to do the good work. So thank you so much. Thank you, everybody, and look forward to the next this thing. Bye-bye.
Thank you. Bye-bye. Nice.
Everybody, bye-bye.
Thank you so much. Yeah.
Thank you, sir. Bye-bye.
Yeah, bye-bye.